Hey everyone, welcome back. We're going deep today into the human genome and aging.
Oh, very cool.
Yeah, and we're going to be looking at this book, Ageless. The new science of getting older without getting old.
Okay.
So, yeah, are you ready to unpack some serious science?
Absolutely, I'm ready.
Okay, great. So remember all that excitement back in 2000 when the Human Genome Project was finished?
Oh, yeah.
Everyone thought we were going to, unlock personalized medicine and predict the future just from our DNA.
Yeah, there was so much hype around that announcement.
Right.
But really all they did was sequence the genome, and that was just the first step. You know what we've learned since then has, totally changed how we think about aging? Our genome is way more complicated than we thought. It's not just a simple book of life.
Okay, so it wasn't as straightforward as we thought.
No, not at all. It's more like, think of it this way. The human genome is like a giant library with tons of books, but only a tiny fraction of those books actually have the instructions for building and maintaining our bodies. Most of the library is filled with, well, we don't know what it's filled with. It's like trying to learn how to build a house, and all you can find are cookbooks.
So what about all those other books? What are they doing there?
That's the really interesting part. Scientists used to call it junk DNA because they thought it was useless.
Right.
But now we're starting to understand that this so-called junk might actually be super important for how our genes work and how we age.
So it's not just about the genes themselves, but how they're controlled.
Exactly. That's where epigenetics comes in. It's like this control panel that sits on top of our genes, deciding which ones are turned on or off.
So even if two people have the same genes, like identical twins, their epigenetics could be different.
Yeah.
Leading to different outcomes.
Precisely. That's why identical twins, even though they share the same DNA, they can end up with different health problems, different personalities, and even different lifespans. Their genes are the same, but the way those genes are expressed can be totally different.
Because of their environment.
Yeah, their environment and their experiences.
So it's not just nature, it's nurture too.
Right.
Our experiences can actually change how our genes work.
Exactly. Think of it like a piano with all the keys. But epigenetics is what determines which keys are played.
Wow.
And in the end, what song is produced.
That's a great analogy. How does this control panel actually work? What makes some genes get turned on and others get turned off?
Well, imagine a landscape with hills and valleys, like a ball rolling down this landscape. And that ball represents a cell developing and specializing. As the ball rolls, it can take different paths, ending up in different valleys. And each valley represents a different cell type, like a skin cell or a brain cell. That's epigenetics at work, guiding the cell's fate.
So all cells start with the same DNA.
Right.
But their journey through this epigenetic landscape leads them to different destinies.
Exactly. And that's what makes John Gurdon's work so groundbreaking. Back in the 1960s, he took the nucleus from an adult frog's intestinal cell and he put it into a frog egg that had its own nucleus removed.
Wait, so he put the brain of one cell into the body of another?
Yeah, basically. And what happened was incredible. That egg developed into a tadpole, a clone of the original frog.
Wow.
This proved that even specialized cells, like those from the intestine, they still have all the genetic information needed to create a whole organism.
So it's possible to reprogram cells?
Yeah.
Back to a younger, more versatile state. Is that how cloning works?
You got it. And then a few decades later, we had the cloning of Dolly the sheep in 1996. The first mammal cloned from an adult cell.
Yeah, Dolly was a rock star.
It was huge.
I remember all the headlines, but I also remember hearing that she had some health problems and didn't live very long. Did that have anything to do with being a clone?
Dolly's case is complicated, but it did raise concerns about the health of cloned animals. But the important thing is that other cloned sheep have lived healthy lives with normal lifespans. So we know that this reprogramming process can work.
So there are still some things to figure out.
Right.
But the possibility of manipulating cells to reverse aging is there.
Yeah.
This is mind-blowing.
Yeah.
So where do stem cells fit into all this?
Stem cells are like the blank slates of our bodies. They can turn into many different types of cells. Even the ones we need for tissue regeneration. They have a lot of potential for repairing damage and maybe even reversing aging.
So unlike most of our cells, stem cells can become almost anything.
Exactly. They have this amazing potential that scientists are trying to use to help people. Think about it. What if we could use stem cells to regenerate damaged heart tissue after a heart attack or repair worn-out joints?
That would be huge.
The possibilities are endless.
That's incredible. It sounds like stem cells could totally change medicine.
Yeah.
But I know there are different types of stem cells, right?
You're right. There are hematopoietic stem cells, which are found in our bone marrow, and they make all the different blood cells. And then there are pluripotent stem cells, which are like the ultimate blank slate. They can become any cell in the body.
Wow, that's amazing. But I've heard there are some ethical issues about where we get those pluripotent stem cells.
You're thinking of embryonic stem cells or ES cells. They come from embryos, which raises some ethical concerns for some people. Right. But there's been another breakthrough that kind of solves this problem.
Really?
Scientists have figured out how to make pluripotent stem cells without using embryos.
Oh, wow.
They're called induced pluripotent stem cells, or iPS cells, and they're made by reprogramming adult cells.
So we can take like a skin cell and turn it into a stem cell?
Yeah.
That could then become a heart cell or a brain cell?
Exactly. It's amazing, right?
That's incredible. Who figured that out?
It was Shinya Yamanaka, a scientist from Japan. He won the Nobel Prize for this discovery. His work totally changed stem cell research.
So we have these amazing tools, stem cells.
Right.
And the ability to reprogram them. But how do we actually control them?
That's where we get into gene regulation.
Okay.
It's the complex system that determines which genes are turned on or off in a cell.
Okay. I'm ready to learn more.
So remember those transcription factors?
Yeah.
They're like the conductors of an orchestra. They bind to specific DNA sequences and tell the cell which genes to express.
So they decide which genes get to play.
Exactly. Think of E. coli bacteria.
Okay.
They only make the enzymes they need to digest lactose when there's actually lactose around.
That's interesting.
It's a simple system for responding to the environment.
So they just know what to do.
Right. Our cells are much more complex, though.
Right.
They have extra layers of control to make sure the right genes are expressed at the right time.
So there are other players in this orchestra besides the conductors.
Yeah. One of the key players is DNA methylation.
What's that?
It's a process that adds little chemical tags called methyl groups to our DNA.
Okay.
These tags can act like mute buttons for genes.
So they silence certain genes.
Exactly. As cells develop, they get these methylation patterns that determine what they become and what they do.
Okay.
And the amazing part is that these patterns can be passed down to new cells when the cells divide.
Oh, wow.
So the specialized functions are maintained.
Sounds like passing down sheet music.
Right.
With instructions on which parts to play.
Exactly. But these patterns aren't permanent. There are enzymes called demethylases that can remove these methyl tags.
So it's not a fixed system?
Nope, it's dynamic. And there's more. We also need to consider histones.
Histones?
Yeah, remember how DNA is really long?
Yeah.
And needs to be packed neatly inside the cell?
Uh-huh.
Well, histones are proteins that help organize and condense DNA.
So they're like the organizers of our DNA library.
Exactly. And just like DNA methylation, histones can be modified too, and these modifications can affect gene expression. One common modification is acetylation, where an acetyl group is added to a histone.
What does that do?
Acetylation tends to open up the DNA structure, making it easier for genes to be expressed. It's like taking a book off the shelf and putting it on a table.
So we've got methylation acting as a mute button and acetylation as an invitation to play.
That's a great way to put it. These modifications are called epigenetic marks.
Okay.
And they're not only crucial for normal development, but they also play a big role in how we age.
This is all coming together.
Yeah.
But how do we actually measure these changes?
That's where Steve Horvath's work comes in. He discovered a link between DNA methylation patterns and age.
Wait, so you can tell how old someone is just by looking at their DNA methylation?
Pretty much. He found specific methylation sites that can predict biological age very accurately.
Wow.
He calls it the epigenetic clock.
So we have a clock inside our cells that tells us how our bodies are aging.
It is. And this clock isn't just about age. It's also linked to our overall health and our risk for age-related diseases.
That's incredible. What about people who seem to age gracefully?
That's a good question.
Are they changing their epigenetic marks somehow?
That's what scientists are trying to figure out. We know that things like diet, exercise, stress, and even pollution can affect our epigenetic patterns.
So our lifestyle choices really do matter.
Absolutely. There's even evidence that these changes can be passed down to our kids.
Wow, so we have more control over aging than we thought.
It seems that way. But if aging is driven by epigenetics, can we truly reverse the process?
That's the big question.
Well, nature actually gives us some clues. Think about what happens at conception. When a sperm fertilizes an egg, the aging clock is reset to 0.
Right. A baby born to a 40-year-old woman is not biologically older.
Exactly. This reset happens because of some amazing mechanisms. First, germline cells, which are the cells that produce sperm and eggs.
Yeah.
They have really good DNA repair mechanisms that protect them from damage.
Okay.
Second, sperm and eggs go through tough selection before fertilization. Only the best ones survive.
Oh, wow.
And finally, and this is the most amazing part, there's the reprogramming of the genome at conception.
Reprogramming.
Yeah.
So like hitting the refresh button.
Yeah. When a sperm and egg fuse the fertilized egg, it goes through a huge erasure of epigenetic marks. It basically wipes the slate clean and starts a new program for the embryo.
That's incredible. Nature knows how to rewind the clock.
It does. Which brings us back to Dolly the sheep.
Right. She was cloned from an adult cell, but her lifespan was shorter, wasn't it?
Yeah, Dolly's case shows us that messing with these processes can have unexpected results.
Okay.
Her telomeres, which are the protective caps on our chromosomes, were too short. And her health problems made people question the long-term effects of cloning.
So reprogramming an adult cell is tricky.
It is. But remember, other cloned animals have lived normal lifespans.
That's true.
So there's still a lot to learn about how to reprogram cells safely.
Okay.
But stem cell research and epigenetics give us a lot of hope.
I can't wait to see what happens next.
Me too. So last time we were talking about stem cell research and epigenetics.
Right, all that cool stuff.
Yeah, and how scientists are starting to really understand how it all works. Now let's talk about some of the specific research that's happening to try to reverse aging.
Okay, yeah, let's get into the details. What's going on in labs right now? Are scientists really close to, like, turning back the clock?
Well, they're definitely working on it.
Wow.
And some of the most promising research involves directly manipulating those epigenetic marks that we discussed.
You mean like the DNA methylation?
Exactly.
The mute buttons?
Right. The ones that can silence gene expression.
So are scientists trying to, like, remove those methyl tags to turn those genes back on?
That's the idea. There's a lot of interest in using compounds that block DNA methylation or histone deacetylation.
Okay.
It's like taking the mute buttons off and letting the instruments play again. The hope is that by removing these marks, we can rejuvenate cells and tissues.
So it's like giving those genes another chance.
Yeah, but it's not that simple.
Right. I was just thinking, wouldn't that mess with how our bodies are supposed to work?
That's a good point. You see, epigenetics is a very delicate balance. It's evolved over a really long time. While those repressive marks might cause aging later in life, they could be protecting us from things like cancer when we're younger.
Oh, so it's not just a simple on-off switch.
Right. Removing them could have bad side effects.
So are there other ways to reverse aging that might be safer?
Yeah, definitely. Remember the Yamanaka factors?
Yeah, the ones that can reprogram cells into stem cells.
Right.
Those are so cool.
Well, scientists are trying to use them in a more targeted way.
Yeah.
Instead of reprogramming the whole body, they're focusing on specific tissues or organs.
Oh, so instead of turning back time for the whole body, they're just giving certain parts a little boost.
Exactly. It's a more controlled approach
Okay.
that could rejuvenate those tissues without the risks of reprogramming the whole body.
Interesting.
And there have already been some promising results in animals.
That's awesome. So it's like targeted therapy, but for aging.
You could say that. And the possibilities are pretty mind-blowing.
I bet. Imagine repairing a damaged heart or regenerating cartilage in a knee.
Or even improving how the brain works.
That would be incredible. It sounds like we're on the edge of a whole new kind of medicine.
I think so. But even if we can figure out the science, what about the ethics?
Oh, yeah, that's a good point. What happens if we can actually slow down or reverse aging? Who gets access to these treatments?
Exactly. Will everyone be able to afford them? Or will it just be for rich people?
That's something we really need to think about.
We do. We don't want to create a world where only some people can live longer.
No, definitely not. And there are other ethical questions, too, like what would happen to society if people could live much longer?
Right. Would it strain our resources?
Yeah, or change how we think about life and death.
These are big questions that we need to start discussing now.
Definitely.
Before these technologies become available to everyone. It's not just about the science, it's about our values.
I agree. But we also have to recognize how far we've come.
We have.
Just a few decades ago, reversing aging was pure science fiction.
It was.
And now it's a real field of research.
With the potential to change everything.
Yeah, it really is amazing. But we're still in the early stages.
Right.
There's still a lot we don't know about aging.
We do need to be careful.
Yes.
And make sure we do our research before we start offering these treatments to people.
But the future of aging research is looking pretty bright.
It is. It really is. So we've talked about some of the exciting research.
Right.
But I'm still curious about those people who just seem to age slower than everyone else.
Me too.
Are there things in their genes or their lifestyle that explain this?
It is a fascinating question, and scientists are definitely looking into that. There are groups of people who seem to age really well, and researchers are trying to figure out why.
Like those communities where everyone lives to be over 100, right?
Yeah, exactly.
They've heard about those places like Okinawa in Japan and Sardinia in Italy. They're called blue zones, right?
Right, and they've become like the perfect places to study longevity. Scientists are looking at everything about them, their genes, their diet, their social lives, their habits.
Everything.
All trying to understand why they live so long. So it's not just one thing, it's a combination of factors.
Yeah, most likely. It could be their diet.
Lots of fruits and vegetables.
Yeah, fresh fruits, vegetables, whole grains. Or it could be that they're active and have close relationships with family and friends.
That makes sense. It sounds like they've found the secret to healthy aging.
They might have. But I wonder if there's something special about their DNA, too.
That's what I was thinking. Do they have genes that make them live longer?
There is definitely a genetic component to longevity. Researchers have found some genes that are more common in people who live to be 100 or older.
Okay.
These genes might be involved in things like DNA repair or how cells respond to stress.
So it's like they have a head start?
Sort of. But even with good genes, lifestyle still matters.
That's true.
Even people who are genetically predisposed to live longer can shorten their lives if they don't take care of themselves.
Right, like smoking or drinking too much.
Exactly. Those things can speed up aging and increase the risk of all those age-related diseases.
So it really is this combination of nature and nurture.
It is. We inherit certain genes, but our choices have a huge impact on how those genes are expressed.
And ultimately how we age.
Right. It's pretty amazing to think that we have some control over the aging process.
Yeah, it is. We can't change our genes, but we can make choices that support healthy aging.
Exactly. Choices that might even help us live longer, healthier lives.
This has been an incredible deep dive.
It has.
We've explored the human genome, epigenetics, stem cell research, and longevity.
So much.
It's amazing how far science has come in understanding aging.
It really is. And we're just getting started.
I know.
As we learn more about aging, we'll probably see even more incredible discoveries.
Imagine a future where we can not only slow down aging, but also regenerate tissues, prevent diseases, and even improve our brain function.
It's an exciting future, but we need to be careful.
Right. We talked about some of the ethical issues.
We did, but it's a conversation that needs to keep happening as science advances.
As we gain the ability to control aging, we need to ask ourselves some important questions.
Yeah, like how do we make sure these technologies are used fairly?
Right. And what does it even mean to age and to live a long life if we can potentially live for hundreds of years?
These are questions for everyone to think about, not just scientists and politicians.
Absolutely. The future of aging is in our hands.
It is.
And we need to shape it wisely. So, to our listeners, as you keep learning about aging and longevity, we'll leave you with this thought. What if scientists do figure out how to safely manipulate epigenetic marks?
That's a big if.
It is. How should we use these technologies? What are the ethical implications? It's something to think about as we go forward.
It's a future full of promise and challenges. We need to be excited about the possibilities, but also aware of the risks.
Well said. Thanks for joining us on this deep dive into the science of aging. It's been a fascinating journey.